Okay, here's something to think about. We share, what, over 98% of our DNA with chimpanzees.
That's right, remarkably close.
And yet, humans seem to be way more vulnerable to certain cancers, especially those really aggressive solid tumors. It feels like a paradox, doesn't it?
It really does. It's one of those big questions in biology.
Well, today we're going to dive into some really groundbreaking science that might actually hold the key to explaining this difference.
Yeah, it's pretty fascinating stuff.
So our mission, as always, is to take your sources, in this case, some incredible new research out of the UC Davis Comprehensive Cancer Center, just published in Nature Communications, and really break it down for you. We want to give you the core insights, the important takeaways, without you having to wade through all the dense scientific language, a shortcut to getting up to speed.
Exactly. And what we're looking at here is, well, it seems tiny, just a small genetic difference, but the implications are just huge. For evolution, yes, but maybe even more importantly, for how we might treat cancer in the future.
Okay, let's get into it. So imagine this. Just one single change, like one letter swapped in our genetic code.
Just one amino acid difference. Tiny.
Right. And that tiny difference compared to chimps might be a major reason we're more susceptible to these aggressive cancers. It sounds almost too simple.
It does, but that's often where the most elegant science lies, doesn't it? The key player here is an immune protein called Fas Ligand. You might see it written as FasL.
FasL, okay.
Now, think of FasL as one of the body's really important weapons. Its main job is to tell damaged or dangerous cells, like cancer cells, to basically self-destruct.
The programmed cell death thing. Apoptosis.
Precisely, apoptosis. It's fundamental. And this is actually how many of our immune cells, including those really advanced CAR T-cell therapies everyone's talking about, actually kill tumor cells. They use FasL.
So FasL is this critical kill switch. What's the specific difference then between ours and the chimpanzee version?
Okay, so here's the core finding. Our vulnerability seems to stem from a single amino acid substitution at one specific spot in the FasL protein position 153.
Position 153. Got it.
In non-human primates, like chimps, they have an amino acid called proline at that spot.
Proline.
But in humans, somewhere along the evolutionary line, that switched to a different one called serine.
Proline to serine, okay.
And this single change, P143S they call it, seems to make our human FasL uniquely vulnerable. It can be cut, basically snipped, and inactivated.
Like leaving a critical weapon exposed to sabotage.
That's a great analogy, yes.
So that obviously begs the question, if our FasL can be cut and disarmed, what's actually doing the cutting?
Ah, good question. The culprit identified in this research is an enzyme called plasmin.
Plasmin. Okay, I've heard of that.
Yeah, it's a type of protease, meaning it cuts other proteins. And here's the kicker. Plasmin levels are often found to be really high in the environment, directly surrounding aggressive solid tumors.
Like which ones?
We're talking about cancers like triple-negative breast cancer, colon cancer, ovarian cancer, some really tough ones. Plasmin is already known to be involved in helping these tumors spread, actually. It breaks down the matrix around cells.
Right, so it helps them metastasize. Okay, so plasmin's already a bad actor in cancer spreading.
Exactly.
And now this research shows it's also disarming one of our immune system's main weapons, FasL, right in the tumor microenvironment.
Precisely. It's a double whammy.
Wow. Okay, this starts to connect some dots then, because we know that some of the most advanced immunotherapies, like CAR T cells, work wonders in blood cancers.
Often very effective there.
But they've been, well, kind of disappointing against many solid tumors. Could this be why? Because the plasmin in those solid tumors is just neutralizing the FasL weapon.
That's exactly the hypothesis this research strongly supports. Blood cancers generally don't rely on plasmin for their spread in the same way solid tumors do. So in blood cancers, FasL can do its job.
But in solid tumors, the tumor's own environment fights back by using plasmin to disable FasL.
Correct. And it gets even more critical because CAR T cells don't just kill the cells they directly target. They also rely on FasL for something called bystander killing.
Bystander killing? What's that?
It means they release FasL, and it can trigger apoptosis in nearby tumor cells, even ones the CAR T cell hasn't directly latched onto. This is super important in tumors that are, a mixed bag of different cell types, some hiding from the main attack.
Ah, so it helps clear out the ones trying to hide or the ones that don't have the main target molecule.
Exactly. It broadens the attack. But if plasmin is there, chopping up the FasL,
then that whole bystander effect just fizzles out.
It's severely compromised, yes. Which could be a major reason these therapies struggle against solid tumors.
Okay, that paints a really clear picture. So this is a compelling idea, But how did the researchers actually, prove this? What experiments did they do?
Well, they did quite a few rigorous tests.
Mhm.
First, they directly showed that human FasL is indeed very easily cut by plasmin,
Mhm.
and specifically at one location between residues arginine 144 and lysine 145.
Okay, very specific.
Yeah.
144 RK 145.
Exactly. Then they compared it to FasLs from rhesus monkeys. Remember, monkeys, like chimps, have that protective proline at position 153. And guess what? The monkey FasL was significantly resistant to being cut by plasmin.
Even though plasmin could still bind to it.
Yes, that's a key point. Both human and monkey FasL could bind plasmin equally well. But that single amino acid difference, the proline versus serine, changes the protein shape just enough, it seems, to protect the cleavage site in monkeys while leaving ours exposed.
Wow. Okay, and what happened functionally? Did cutting the human fat cell actually stop it from working?
Absolutely. When they treated human FasL with plasmin, it completely lost its ability to kill cells. It also failed to activate a key molecule downstream in the death pathway called caspase-8.
Okay.
And to nail it down, they actually engineered a mutant human FasL where they changed that specific 144RK145 cutting site.
Ah, so plasmin couldn't cut it anymore.
Exactly. And that mutant protected version, it worked just fine, even with plasmin around.
That's pretty definitive.
Yeah.
Did they look at other animals too?
They did look at mouse FasL. Interestingly, mouse FasL can be cut by plasmin, but at a totally different site, much further away from the critical functional parts compared to where human FasL gets cut.
So this specific vulnerability at 144 RK145, linked to the serine at 153, seems quite uniquely human, or at least primate-specific in this detrimental way.
It certainly appears that way based on this work, yeah.
They also mentioned something about aggregation, how FasL needs to clump together.
That's right. For FasL to effectively trigger apoptosis, it needs to form larger clusters or aggregates on the cell surface. The researchers showed that the plasmin-cut, shorter version of human FasL was terrible at forming these aggregates.
So it gets cut, it can't clump together properly, and therefore it can't give the kill signal effectively.
You've got it. The damage prevents the necessary structure formation.
Okay, so we've identified the problem. Plasmin in tumors disabling our FasL weapon. Did the research offer any solutions? How can we, rearm our immune system?
Yes, absolutely. And this is where it gets really exciting therapeutically. The study showed two main strategies could work.
No.
One, you could block plasmin itself. Use plasmin inhibitors, like a drug called aprotinin, for example, to just stop it from being active in the tumor environment.
Right, take out the saboteur.
Exactly. Or, the second approach, you could specifically shield the FasL protein from being cut without stopping it from working.
How would you do that?
This is where they develop some really clever tools.
Yeah.
Specific antibodies. Think of them like tiny molecular bodyguards for FasL.
Antibodies, like Nuc2, No2-Ecker, 9F5, those names they mentioned?
Precisely those. These antibodies are designed to bind to the stalk region of the FasL protein right near the cleavage site.
Okay.
So they physically block plasmin from getting access to cut it. But crucially, they don't interfere with the part of FasL that needs to bind to the cancer cell receptor to trigger death.
So it's like putting a protective cap on the vulnerable part, but the business end can still do its job.
That's a perfect way to put it, yes.
And did this work in practice, in their models?
It worked remarkably well in their preclinical models. When they used these protective antibodies, they saw that CAR T cells could effectively carry out that crucial bystander killing again, even in environments loaded with plasmin.
Wow.
They saw the downstream signals like caspase-8 activation and another marker called cleaved caspase-3 get restored. And importantly, when they combined these antibodies with an existing immunotherapy, an anti-PD-L1 drug.
Like Evolutionary.
Yes, like avelumab. When they combined them, they saw a significant reduction in tumor size. And this was even in really challenging models like patient-derived ovarian cancer cells that were already resistant to chemotherapy.
That's incredibly promising. Shielding FasL could potentially make existing immunotherapies work much better against these tough solid tumors.
That's the hope, absolutely. It opens up a whole new angle of attack.
Okay, the therapeutic side is huge. But let's circle back to the evolutionary aspect because that's just mind-bending. Why would humans evolve this apparent vulnerability? It seems counterintuitive for survival.
It does, doesn't it? And the paper offers a really intriguing hypothesis, though it's still a hypothesis at this stage.
Okay, what is it?
They suggest that this specific mutation in FasL, the switch from proline to serine at position 153, might have actually played a role in another key human trait: are exceptionally large brains.
Our brains. How?
Well, it turns out FasL is also involved in development. Specifically, during embryonic brain development, it helps regulate the number of neural progenitor cells through programmed cell death.
Ah, so it prunes back some developing brain cells.
Exactly. The idea is that maybe the human version of FasL, the one with serine, is slightly less efficient at triggering the cell death during brain development compared to the primate version with proline.
So slightly less pruning.
Could have led to more surviving neural progenitors, contributing to the expansion of the cerebral cortex, the thinking part of our brain.
Wow. So the mutation that makes us vulnerable to cancer might have been advantageous for developing bigger brains. An evolutionary trade-off.
That's the fascinating possibility they raise. What was beneficial for cognitive evolution, a larger brain might have left us with this, as they call it, unfavorable concession when it comes to cancer susceptibility in these specific plasmin-rich environments later in life.
That reframes everything, doesn't it? Evolution isn't necessarily about perfection, it's about trade-offs. Something good for one thing might have a downside elsewhere.
Precisely. It's not linear progress, it's a complex balancing act. And we see hints of this elsewhere in genetics, too, like with genes such as Ki53 or BRCA2, where variations linked to cancer pathways seem to have undergone different evolutionary pressures in humans versus chimps.
It makes you wonder what other hidden trade-offs are written into our biology from our evolutionary past.
Absolutely.
So just to recap for everyone listening, we've journeyed through this incredible discovery showing how a tiny, ancient change in our Fas Ligand protein, possibly linked to brain development, makes human FasL vulnerable to being disabled by plasmin in solid tumors.
Yeah, a vulnerability our primate relatives don't seem to share in the same way.
And understanding this deep evolutionary quirk is now paving the way for really innovative strategies, like those protective antibodies, to potentially boost cancer immunotherapy for solid tumors.
It really challenges us, doesn't it, to think about biology not just in the present, but across evolutionary time. How adaptations from millions of years ago might be influencing our health challenges today. It makes you ask, what other complex evolutionary stories, these sort of unfavorable concessions, are waiting to be uncovered in our genome?
It's a profound question, and hopefully for you listening, this deep dive highlights just how powerful this kind of evolutionary perspective can be. Understanding our past might be one of the most crucial keys to unlocking better health and more effective treatments like personalized medicine for the future. A lot to think about there.